Hello to all students. In this video lecture, we are going to discuss genetic code. As you know that all human beings, animals and plants, their bodies contain proteins. Their bodies are made up of proteins, especially of animals. In human, there are 10,000 different types of proteins. These proteins include like hair, nail, muscles, enzymes, hormones, hemoglobin, etc. These proteins are synthesized by the instructions from our DNA, which are present in the chromosomes. These instructions are carried in the form of genetic code or simply codon from DNA in the form of messenger RNA to the ribosome, where proteins of different types of proteins are synthesized according to that genetic codes present on messenger RNA. So it is very important because a small or little mistake in the genetic code or codon can lead to serious consequences which can lead to different diseases. For example, in the synthesis of hemoglobin, there are 574 amino acids involved in the form of four chains, 2 alpha chains and 2 beta chains. And in a single mistake in the base pair sequence in the genetic code can alter the structure of hemoglobin, which can lead to the sickle cell anemia and can lead to the death of the person. So these codes are very important for the existence of a living organism. So what is a genetic code? Genetic code is a 3 nucleotide base sequence. It's like triplet code. There are different bases present on DNA like adenine, guanine, cytosine and thymine. So we are going to take 3 nucleotide base sequence. In messenger RNA, because we are referring at a process known as translation, which is very important for the protein synthesis, and messenger RNA is taking the message from DNA, complementary base sequences from DNA in the form of messenger RNA. So that's why we are considering messenger RNA over here, because 3 nucleotide sequence present on messenger RNA, mRNA, that act as a code word for amino acid, for a particular amino acid in protein. And these particular amino acids are joined in a specific number and in a specific sequence to synthesize a particular type of protein. So as you know, there are 4 types of nucleotides in DNA: adenine, guanine, cytosine and thymine, and their complementary base pair present. 4 types of base pairs are present on RNA, which are adenine, guanine, cytosine and uracil, A, G, C, U. So why there is a triplet codon? Why it's not only 1 base pair or 2 base pair? If we include 1 base pair, so it will make only 4 codons, but there are 20 different types of amino acids which join together to form proteins. So these 4 codons are not enough for 20 types of amino acids. So if there are 2 types of base pairs like AG, GC or CU, then there are total number of 16 codons, which are also not enough for 20 different types of amino acids. So that is why nature has decided to take 3 base pairs together to form a genetic code or codon. So when we talk about 4 power 3, then it is equal to 64. So 64 codons are more than enough for 20 different types of amino acids, which are essential for the synthesis of different types of proteins. Now how these genetic code or codons are formed? So we have to make a table for that. This table is made up of 16 boxes. So as you can see in this diagram, there are 16 chambers or boxes, 1, 2, 3 and 4 right over here. 1, 2, 3 and 4. So total number of boxes are 16. So we are going to put 4 different types of nucleotide present in messenger RNA in a specific sequence right over here. U in the first box, C, A and G. So these are the base pairs which start from 5' end of messenger RNA. So this is the first base which is present on the messenger RNA. The second base, the middle one, also can be written on the top of the boxes in the same sequence like U, C, A, G. So you have to remember this sequence U, C, A, G, U, C, A, G, this side and on the top. While the third nucleotide or third base pair, which is going towards 3' end of the messenger RNA, is going to be written on this side. But at this level, each box contains all those 4 base pairs like UCAG in this box, UCAG in this box, and same over here and same over here. So once you have drawn all these boxes along with the base pair, then it is very easier for you to make a genetic code. So how you gonna do it? First of all, take a base from this side, which is U, then from this side, which is also U, and the first base from this side, which is also U. So the first code will be UUU. In the same manner, you can also synthesize second code, U from here, and now we are going to take C, cytosine from here. So it will become UUC. In the same manner, UUA, UUG. Now these two genetic codons which represent an amino acid leucine. In the same manner, we can synthesize genetic code right over here. We are going to take C from this side, C from this side, and one by one all these 4 base pair from this side. So this will become CCU, CCC, CCA, and CCG, which all these 4 genetic codes or codons represent proline, which is an amino acid. If we look at on this box, so we are going to take G from this side, go upside, G from this side, and then one by one bases from this side, UCAG, one by one. So it will become GGU, GGC, GGA, and GGG. So these 4 genetic codons represent glycine, which is also an amino acid. So in this way, you can synthesize all 64 codons in a very easy manner. Out of these 64 codons, one codon which is right over here, AUG, which is represent methionine, it is known as start codon because this codon is always present on the at the start point of messenger RNA to synthesize a particular protein. While UAA, UAG, and UGA right over here, these are known as stop codons because they are present at the terminal portion of messenger RNA to stop the synthesis of protein. So that is why these 3 codons are known as termination codons, stop codons, or nonsense codons. Now we are going to discuss these properties of genetic codons one by one. First of all, I'm going to repeat termination codons. There are 3 termination codons, UAA, which is known as amber, UAG, which is known as ochre, and UGA, which is known as opal. So these are 3 codons which are nonsense codons, stop codons, because these codons do not represent any amino acid. Rather, they are used for the stop signal for the synthesis of a particular protein, and they are present at the terminal portion of messenger RNA. Start codon is always AUG, which represent methionine, and it is always present on the starting point of messenger RNA, which initiate the process of synthesis. If we look at the properties of genetic codon, genetic codes are universal. All codons are universal in all organisms, animals, humans, and plants. But there are some exceptions. For example, a codon which is known as AUA, if this codon is present in mitochondria, it will represent methionine amino acid. But at the same time, if AUA is present in cytoplasm, then this genetic code will represent isoleucine, which is second type of amino acids. So there are exceptions. Otherwise, these exceptions, all organisms contain similar types of genetic codons which represent similar types of amino acids. They are very specific. If we look at their specificity, they are always specific for same amino acid. If we look at right over here, GGU, GGC, they are always represent glycine. If you look at here, ACU, ACC, ACA, ACG, they all always represent threonine, which is amino acid. So each genetic code is always specific for a specific amino acid. They are non-overlapping. It means that they can be read continuously without any comma, full stop or any other kind of mark. Like there is a genetic code UUU, CUU, AGA, GGG. So we are going to read it continuously without any comma or punctuation. So there are 4 genetic codons right over here. It means these codons can only produce a protein which contain only 4 amino acids. So if the protein is larger with many amino acids, then the genetic code will also be a longer one. And if the protein is shorter or the smaller in size, then it also contain less amount of genetic codes or codons on the messenger RNA. Genetic codes are also redundant. As you know that there are 64 codons, but out of 64 codons, 3 codons are nonsense codons, stop codon, termination codons. So there are only 61 codons left which represent a particular amino acids. So 61 codons are used for 20 different types of amino acids in our body. For example, glycine has 4 codons. And if you look at this diagram, this table, GGU, GGC, GGA, GGG, they all 3 codons represent a single amino acid which is glycine. So these codons are known as synonyms. So there are different types of synonyms present in this table. You can see right over here, these codons represent threonine, these codons represent proline, these codons represent serine. So these are known as synonyms. So that's all for today. Hopefully it makes sense, and I'll see you in the next lecture. Until then, bye.